Looks like we are getting ready to start here. Well, welcome everyone. I am Terry Deshanes Lamb with the Healthcare Partnerships team at MDA. Thank you for joining us for today's MDA Industry Update webinar presented by Edgewise Chief Scientific Officer and co-founder Alan Russell and Edgewise Therapeutics Chief Medical Officer Joanne Donovan. MDA's Industry Update webinars are just a part of MDA's community education programs. We encourage you to visit the community education pages of mda.org. There you'll find an abundance of resources, informative recorded webinars, and listings for upcoming educational events. And of course, if you have any questions or need assistance, the MDA Resource Center is available to you by email at resourcecenter@mdausa.org, calling 1-833-ASK-MDA1, or you can reach the Resource Center on mda.org. Now, I'd like to turn it over to Edgewise Therapeutics Chief Scientific Officer, Alan Russell, for today's presentation. Dr. Russell, you can take it away from here. I'm going to stop sharing.
Thank you very much.
You're welcome.
Yes. Should start this. Yeah. Hopefully you guys can see that. Look okay? Uh-oh.
Looks great.
It's got a life of its own, but I think I'm back. So thank you very much for the opportunity to talk about the program. I'm Alan Russell. I'm the Chief Scientific Officer of Edgewise. I'm based in Boulder, Colorado, and I'll introduce Joanne Donovan, our wonderful MD lead for the program in the second half of the talk. And we're here to talk about 5506, what it does and what it can do for you, hopefully, in some of the trials that we're setting up now in both Becker and Duchenne muscular dystrophy. Let's hope this advances. Yes, of course, we're a public company, so I have to put one of these forward-looking statements up first, just to really make one point that the compound that we're talking about, EDG-5506, is an investigational compound, and that means it's not approved for any indication anywhere at this point. It's in trials, and so we can't make any specific promises about what it's going to do for you. We can tell you what it does, what we hope it does for you, but we have to put this warning in front of everything. And this is just a reminder to me, just to tell you a little bit about the company. We were founded in 2017, about 5.5 years ago, based in Boulder, Colorado, but we're largely split between there and Boston. And really the founding goal for the company was to develop 5506 and to try a different way of treating muscular dystrophies that was more practical, had low side effects, you could take it orally, and was amenable to people of every genetic mutation within the dystrophinopathies, and also potentially combinable with stuff. So it was really a goal to develop a medicine that was practical and useful for you folks out in the community and clinic. I got this slide from our advocacy experts, Abby and Catherine, and it's really just, before I start going on about the science, just to say how we view advocacy at Edgewise, because it really is core to how we work. And there's two main parts to what we do within the advocacy realm. And the first is to listen to you guys. And we do that through patient-centric focus groups. We also do that by attending community events. And you'll see our presence at nearly everything that we can possibly attend, hopefully to get our faces out there and to get you more familiar with the story and what we hope to do. In listening, what we're really hoping to achieve is to improve how we do our clinical trials. We want to make it as easy as possible for you folks, and we want to measure things that are relevant to the problems that you face. So we can only really do that by listening. So that's really our primary goal. And the second part of our interest is really to help the advocacy, patient, stakeholder community in helping to understand gaps towards improving standard of care in the treatment landscape for you folks. right? So we do everything that we can to join that kind of community to get that voice out of the unmet need in the community and how to improve it. I'll now tell you about some science, hopefully. So I'll just introduce the kind of high-level aspects of 5506, and then I'll tell you about what it does. So it's important to understand how dystrophin works, and I'll tell you about that. There's three main aspects to our message in the kind of preclinical realm. The first is that some muscle fibers are more susceptible to damage due to the lack of functional dystrophin. Our compound selectively targets the fast fibers in your body that break down more readily, and I'll tell you a little bit more about that as we go forwards. As I mentioned, we've made this investigational therapy, EDG-5506. It's designed to protect those fast fibers from damage, regardless of the dystrophin status. So whether you're a Becker or a Duchenne, it's really a structural fix to the problems that these muscles face rather than a genetic fix. And then in diseased animal models, so these are largely models of Duchenne, although we have done some work in a model of Becker as well, 5506 protects susceptible muscle fibers and prevents long-term development of damage. So when the muscle contracts, it's much less injured. And I'll show you a graphic of that in the next slide or two. So before we get to what the compound does, I just wanted to give you an overview of what dystrophin does, right? Often you see pictures kind of like that thing on the left, kind of a static image, but of course muscle contracts. And on the right-hand side, here's the muscle contracting as we like to think of it. The dystrophin is contained in those green stripes connecting the muscle together. It's part of a large complex of muscle, of proteins, and it helps those fibers stay together. Now, muscle fibers don't always contract at the same time. And in this case, what you're seeing is the central fiber contracting, but not the other ones. And you can see that those green stripes are helping keep everything together. So if those things aren't present, the body tries to upregulate some proteins to make do. That's the blue stuff. They don't work very well. The end product is really that when a muscle fiber contracts and you don't have functional dystrophin, the fiber is on its own. It can contract perfectly normally, but it's much more likely to suffer stress when it does so because it doesn't have all of its friends around it supporting the contraction. And when muscle fibers get stressed, what tends to happen is the membrane gets upset, and that causes further hypercontraction and breakdown of the fibers, and you'll see that about to happen now. And that's largely because you have activation of proteins within the muscle fibers that cause it to kind of chop up. There you go, bang. So you're actually activating proteolytic enzymes that almost apply scissors to the muscle fiber to get rid of it. It happens in you and I when we go to the gym and do exercise, and it's how we adapt our muscle to get stronger. But of course, if you have Becker or Duchenne and the dystrophin complex is not fully functional, then instead of an adaptive process, it becomes a disease process where too many of the muscle fibers are breaking down, and that leads to the inflammation and then the fibrosis and the muscle replacement that causes the functional declines with age. This is just the same thing again in graphical form in case my video didn't work. But you can see here that where you have, I'll just turn my pointer on, where you have this central fiber contracting, these other fibers, when they're connected together through the dystroglycan complex, can help support contraction of that fiber. And if that element is not present properly, then that fiber is going to get more stress. And our goal is to decrease the stress on these individual fibers by applying 5506 and modifying how the muscle fiber contracts. And here is everything all at once. So hopefully these videos will move. Yep. So on the left-hand side, this is a control compound from a Duchenne mouse. So this is the MDX mouse. It has no dystrophin. And when the muscle contracts, it breaks down. And you can see this happening here. You see this sharp line here. These are these enzymes that are being activated from the stressed muscle fibers, chopping up the muscle And then what happens is it retreats into these blobs, which are essentially hypercontracted muscle fibers, ready to be disposed of, and new fibers grow. This is because there's no dystrophin in this muscle, so when these fibers contract, they get excess stress, right? On the right-hand side, we pre-incubated the muscle with the compound 5506 for 1 hour. And what 5506 does, it targets the myosin in the fast fibers of this muscle, which makes up nearly all of this muscle, to change how that muscle fiber contracts. It can still contract as a normal muscle does, but it puts a speed limiter on the extent of contraction. So we've just turned down the contraction by about 15% on this muscle to limit how much it can injure itself. So as a speed limiter on this muscle, it's still fully functional. It just can't push itself to the breakdown point that you see on the left-hand side. So it's contracting, but now what we're seeing is it's no longer falling apart. As I mentioned, we target the fast fibers in the body. That's about 50% of our muscle makeup, and those muscle fibers are more susceptible to this type of injury than the slow ones. Okay, so we target half of our skeletal muscle with a goal towards changing how it functions and how it breaks down. It shouldn't limit physical function, but it should limit disease function such that you should be able to use your muscles in a healthy way, but not in a damaging way. Now, Joanne will tell you about the clinical data shortly. I just wanted to give you some context of what kind of biomarker stuff that she'll show you. So most of you are familiar with measuring creatine kinase, or CK. So CK leaks out of muscle when muscle fibers are injured. So in that video, when you saw that collapse of that muscle fiber, that's the type of event that causes the release of proteins from the muscle fiber into the bloodstream. Now, we're all familiar with creatine kinase. It's often how Becker or Duchenne were initially identified. But of course, that's not the only protein that leaks out of these large cells. We also see things like myoglobin, another protein enriched in muscle fibers. And I'll tell you a little bit about fast troponin. That's another structural protein in these muscle fibers that leaks out, and lots of other stuff as well that we measure. But really, what we're interested in is seeing an aggregate measure, and we tend to measure more than just creatine kinase, and I'll just tell you about fast troponin. So here's creatine kinase in a cross-section of Duchenne, Becker, or control individuals. And you can see, as you might expect, healthy people have low levels of creatine kinase in their blood, Becker in the middle, and Duchenne in the right there have the higher levels. So Becker in the middle, they have medium levels, but above control, and then Duchenne have high. The same is also true of this other protein that leaks out, fast troponin. Now, the only difference really is that fast troponin is only expressed in fast fibers. Now, if you remember, that's the target for 5506, making up half of our skeletal muscle. So this is a biomarker we view as really it gives you an indication of how well the compound is working, because this one is specific to the fast fibers, whereas creatine kinase comes from all sorts of tissues and muscle, okay? And you can see high levels in Duchenne, medium levels in Becker, and low levels in control, right? So I just wanted to give you that context because Joanne will be showing you some fast troponin data in a little. And just one extra preclinical piece of information that helps frame the Becker data that we'll be showing you. We did do some dosing of 5506 in probably the best model of Duchenne. That's the golden retrievers with muscular dystrophy. So they're a large dog model of muscular dystrophy. They're a natural model that was identified in the community. These dogs tend to get sick around 1 to 2 months of age, and then their disease progresses till about 6 months of age, where they stabilize. We actually took dogs that were stabilized, so they're about 7 months of age, so they're already developed symptoms of muscular dystrophy, and we gave them a short period of dosing orally with 5506, and we observed two things. The first thing was that the CK dropped about 50 to 60% in those dogs during the dosing period, and then when we took the compound away, the CK went back up again. So this is very consistent we're decreasing the leak of the muscle. And then at the same time, what we noticed was the dogs were actually more active. We used a collar-bound activity monitor. So it's from the pet store type of activity monitor. You can measure how active the dogs were. And instead of being less active, they were actually more active. So even though we were limiting their muscle contraction, we were actually observing that they were more active because the muscles are no longer breaking down as much, right? So this is kind of a very useful frame as we move into the clinic as the kind of things that we might see in people. At this point, I'm going to turn it over to Joanne, and she'll tell you a little bit about the treatment landscape as we see it, and then some specific data in the trials that we've done to date.
Hello there. I'm delighted that MDA gave us the opportunity to speak with you today. I'm Chief Medical Officer at Edgewise, and we are studying both Becker and Duchenne muscular dystrophies. Now, if you're interested in Becker, why do you care about Duchenne and vice versa? It's because that same process of the lack of dystrophin, either complete in Duchenne or partial in Becker, the lack of dystrophin doesn't allow the muscles to protect themselves during everyday activity. So we are looking at both Becker and Duchenne, and that actually gives us more information across the spectrum of disease. So in the next slide, we started in a phase 1 study. That's the first study that you look at in people. And basically, we looked in unaffected adults as well as a number of men with Becker muscular dystrophy. And the first thing, of course, you want to know about safety. And we saw that we dosed it for 2 weeks. It was well tolerated. This is a tablet. So EDG-5506 is given as an oral tablet once a day. And we found that it's absorbed well, either with or without food. And a very important question in developing a drug is it getting to where it needs to be? And we found that EDG-5506 is not just in the bloodstream, but it's 100 times that concentration in muscle. We did muscle biopsies in the unaffected adults as well as those folks with Becker muscular dystrophy. And I do need to give a shout out to these guys because they stayed in a phase 1 unit, which is not the most pleasant place, for 2.5 weeks. And we can't thank them enough for their contributions here. On the next slide, what did we see? And Alan has told you about the biomarkers of muscle damage that we see both in Duchenne and Becker. CK, we saw that was decreased almost 40%. It happened early and it was sustained now at 6 months. These folks have been on the drug now for more than 6 months, a dozen men. And we also saw that the fast skeletal muscle troponin, the very specific biomarker that tells us we are targeting the protein we wanted to target, is down 75%. So what this is basically saying is that if you can prevent the damage that occurs with everyday activity, then can that translate into an improvement in function? And we can't really say that with 10 patients for sure, 12 patients. What we can say is we looked at the North Star ambulatory assessment in these. That's the same North Star that is used in children with Duchenne. And on average, these men had fairly advanced Becker, kind of comparable to what happens in maybe 10 to 12-year-old boys in terms of their function, those with Duchenne. And we saw actually that the North Star ambulatory assessment trended upwards. And in these men, we would have expected from what we know of the natural history of the disease that they would have gone down somewhat. So that's encouraging. We also saw this in a different scale that is more oriented towards limb girdle type muscular dystrophies. And again, we saw that improve. And that's an indication also we're thinking about other types of muscular dystrophy beyond Duchenne, and Becker. So positive trends here, even in a limited period of time. So what that gave us the basis for was several clinical trials that are ongoing, and we are certainly planning additional ones in the future. The 12-month, now 24-month open label study continues on, and that's adults with Becker, some of whom who were in the phase 1 trial, and some additional folks who were enrolled in that ARCH study. The Canyon study is enrolling now at multiple sites across the United States, and will also be enrolling in the UK and the Netherlands. That is in ambulatory adolescents, so age 12 and up, as well as adult men with Becker. Those are the numbers to look up on clinicaltrials.gov, and I'll tell you a little bit more about these studies. And lastly, an initial study in a phase 2 study in ambulatory boys with Duchenne is also enrolling at several sites now, more to come in the United States, and that's the LINK study. So to tell you more about the LINK study in boys with Duchenne, this is a year-long study. It's a 2-part study with an initial placebo-controlled part, followed by an open label part. And it's really to identify doses in boys that we can look at in a longer term study. Because the drug seems to decrease biomarkers very quickly, we can choose doses, we can understand how it's metabolized in boys relatively quickly, and then move on to a larger study. So that is enrolling boys aged 4 to 9 up to their 10th birthday with a genetic diagnosis of Duchenne that are ambulatory on a stable dose of corticosteroids, and they can also be on a stable dose of exon skipping therapy. And that speaks to, we see this as something that could be valuable on top of steroids, on top of exon skipping, on top of other drugs that are in development as well. And we have sites enrolling. We're going to have more late in the early part of this year. So on the next slide, we are using a 3-month treatment period, placebo-controlled, and then a much longer open label period to understand safety and how the drug is absorbed and metabolized in these boys. We are also using ways on the next slide also of easing the travel burden in these boys. We do have 10 site visits over the 12-month period, including the screening, and we do not have a muscle biopsy in this study. We're looking at monitoring with heart, EKGs, with echoes, also lung function tests and strength tests, as well as the functional measures like North Star that you're used to seeing in the studies. We are drawing blood at home with the Tasso blood collection device, which is a relatively easy way of being able to collect blood for understanding how much drug is in the bloodstream. And that's something that we can do and avoid visits to the clinic. We're also doing monitoring of function with an Apple device, Apple Watch-like device, to look at activity and understand more about the effects of the drug at home. So we are trying to, looking at the broader view of how we develop this drug and planning down the road for expanded age ranges, broader function, including non-ambulatory, and looking for our next set of trials, a broader geographic area and more sites. And we are, as much as we can, developing home-based measures. So, as I mentioned, we're thinking of this in combination with other therapies as well as alone. So, to summarize, we are really committed to reducing the trial burden as much as we can. by listening to you and interested in your questions and comments. To summarize in general, we are looking at the EDG-5506 being developed for Becker and Duchenne muscular dystrophy. On the next slide, it's taken orally. It's intended to preserve and hopefully to improve function in Becker and Duchenne. The goal is to protect muscle, to prevent damage by protecting the most susceptible muscle fibers alone or in combination, and really to stop the damage where it begins, the fundamental problem with the lack of dystrophin in Duchenne and Becker. So thank you for your attention. There on the next, we have the place where we can be reached, also on clinicaltrials.gov. The links are there to reach us and get you in touch with the sites that are currently enrolling in the study. I will say, because I see questions in the box, we are at this point looking at ambulatory patients with Becker. And we are looking at ambulatory patients because we are looking at endpoints like the North Star that we can measure change over a period of time to be able to get the drug approved in the United States and elsewhere. So similar to the Duchenne studies where different groups are looked at in different studies, we are starting with ambulatory. We'll look to expand to non-ambulatory as well, because that is clearly what we want to avoid in Becker as well. So,
John, we have a specific question about the University of Iowa.
Iowa is enrolling adult and adolescent patients with Becker. And also will be enrolling patients with Duchenne as well. So Dr. Matthews is involved.
And we have one question around can a 13-year-old Becker boy join Canyon after they're on high-dose steroids?
So in the Duchenne study, we're looking at boys on steroids. In the Becker, we are looking at adolescents and men who are not on steroids. And again, that is not because we don't think the drug could help in those populations, but from a clinical trial point of view to try to understand the effects of one thing at a time. That's why we are. But ultimately, that's one of those ways where the crosstalk between trials helps, because if we show in Becker that it's useful without steroids, we show in Duchenne it's useful with steroids. That tells us more about how to use it, ultimately how physicians can use it in the long term.
And we had a question around the age range of patients in the ARCH study for the data that you showed.
Those are all adults. Their average age is in the mid-30s, those men.
And a question from Naman. 5506 seems not a gene therapy, safe to say it works regardless of genetic defect? Absolutely. That's the whole goal.
Yeah.
I don't know about you, but it drives me completely crazy how complicated it is that there's so many different genetic variants of both Becker and Duchenne. And of course, it makes a huge difference in terms of antisense oligos and also even whether you can have a gene therapy. Our intervention changes how muscle contracts, not how it's genetically made up. So it should be just fine whether you have Duchenne Becker or Duchenne that looks like Becker or Becker that looks like Duchenne, or even as we go further into the clinic, we'll be testing this in other forms of limb-girdle muscular dystrophy. So it's kind of a fix-all, if you like. Do we have any other questions? Interested in Canyon study for our 17-year-old son with Becker, or you are front and center on the adolescent side. So,
yeah, yes, so that they would be potential. We are opening more sites, so keep an eye on clinicaltrials.gov for that as well. And of course, what I didn't mention.
There was a question around sites in Florida.
Yeah, and what I didn't mention was that travel is provided, prepaid cards and support for travel to minimize the burden on patients and family members.
People always ask how long before this gets to market, but I unfortunately can't tell you that because we have to do the studies first, which is really key. You know, we need to recruit people to these studies as quickly as we can to get the information that we need to share with the FDA.
So if you have more questions, please reach out, and thank you so much for listening and taking the time today.
Thank you so much.
Yeah, wow. Thank you, Dr. Donovan, Dr. Russell. It's MDA's pleasure. And I think it's wonderful to hear such encouraging news in your research. I think everybody on this call will definitely agree. I do want to thank all of our attendees as well and let you know that the webinar has been recorded and will be uploaded to YouTube for on-demand viewing. So please be on the lookout for an email with the viewing information in the next couple of weeks. Also, at the end of this webinar, you'll receive a survey link. And if you could please take the time to complete this short survey so that your voice is heard and you can help us create more impactful programming for future events such as the one that you just saw. Otherwise, I think that concludes our webinar. And I just again want to thank everybody, our presenters, and all of our attendees. And please have a great day. Thank you.